Hello everyone and welcome you all to Quick Solutions for All India Akash Test Series Medical 2020. Today I am going to take up the zoology part of the paper, test paper number 4, code A and B in zoology. So let's get started with the very first question. Question number 136, a native plasmid is, and the options given are, the very first option says, autonomously replicating closed circular extrachromosomal DNA. And this is the correct answer to our question because a native plasmid is an autonomously replicating closed circular extrachromosomal DNA that lack any vital gene. The next option given is single-stranded chromosomal DNA is incorrect because native plasmid is usually double-stranded chromosomal DNA. And this option is incorrect because native plasmid is not a single-stranded chromosomal DNA but an extra chromosomal DNA. And the next option is autonomously replicating vital gene coding DNA is incorrect because native plasmid lack vital genes. And the last option says antibiotic resistance gene containing chromosomal DNA is again incorrect. It is an extra chromosomal DNA. So the correct answer to this question is option number 1. Let's move on to the next question. Question number 137, select a palindromic sequence. Now, a palindromic sequence of a DNA sequence is a sequence of base pairs that read same on the two strands of the DNA when orientation of reading frame is kept same. So you can say there are two mirror images that are present in opposite orientation or direction. So we will look for the sequence in the opposite strands and the direction orientation. In the very first option, the direction from 5' to 3' end reads GAATTC to 3' end. And on the opposite strand to 5' to 3' end, it is GAATTC. So as it is a palindromic sequence, this is a correct answer to our question. The next option, as it has both the strands in the same direction, 5' to 3', so this cannot be a palindromic sequence. The next option is option number 3, from 5' to 3' in the opposite directions. The sequence is not same, so this cannot be a palindromic sequence. And same is with the last option, option number 4, 5' to 3', the sequence doesn't read the same. So this cannot be a correct answer. So the correct answer to this question is option number 1. Let's move on to the next question. Question number 138. In Eco R1, the letter R is derived from the, and the options given are name of the genus, name of the viral strain, name of the bacterial strain, and the last option says order of isolation of bacterial strain. And the correct answer to this question is option number 3, name of the bacterial strain. Because in Eco R1, this E is derived from the genus Escherichia, whereas CO from the name of the species coli. R represents the bacterial strain and not the viral strain, while the Roman number indicates the order of isolation. Order of isolation from that strain of bacteria. So option number 1, name of the genus is incorrect. Option number 2, name of the viral strain is also incorrect because it represents the name of the bacterial strain. And option number 4, order of isolation of bacterial strain is also incorrect. So the correct answer to this question is option number 3. Let's proceed to the next question. Question number 139. The stickiness of the ends of overhanging nucleotide stretches facilitates the action of the enzymes categorized as, and the options given are lyases, DNA ligases, restriction endonucleases, DNA polymerases. And the correct answer to this question is option number 2, DNA ligases. The very first option, lysis, is incorrect because lysis catalyzes the breakdown of substrate into two parts without the use of water. The next option, restriction endonucleases, is also incorrect because action of restriction endonucleases produces sticky ends. And the last option, DNA polymerases, is also incorrect. So the correct answer to this question is option number 2. Let's proceed to the next question. Question number 140. A circular plasmid was taken and subjected to a restriction digestion with multiple restriction enzymes. The resultant mixture was subjected to agarose gel electrophoresis. Observe the figure given below and choose the incorrect statement. In the figure, 4 lanes are given, 1, 2, 3 and 4. These are the wells where you load your DNA sample, and as the gel electrophoresis happens, the DNA fragment separates according to their size. The smallest one will be closer to the anode, and there you will find the smallest DNA fragment, and here you will find the largest DNA fragment. Accordingly, we will see the options. The very first option says possibly completely digested sample is in lane 4. And if you will see in lane 4, the DNA is separated very well in this lane, and you can see the smallest DNA fragment at the very end of it. So this is possibly a correct statement, and so this cannot be a correct answer. The next statement says largest DNA fragment is closest to the wells, and you can see that it is closest to the well. So this is also correct statement, so cannot be a correct answer. Third statement says undigested DNA sample is in lane 2, but in lane 2 also we see there are digested products. So this cannot be a correct statement and is a correct answer to our question. The last statement says smallest DNA fragment is closest to anode. Here you have anode, and here you have cathode. So as it is also a correct statement, this cannot be a correct answer to our question because the undigested DNA sample would be in lane 1. So the correct answer to this question is option number 3. Let's proceed to the next question.